Thank you all so much. Um, Before I jump in, I just want to extend a big thank you to Aiden for inviting me. And also thank you to everyone who's met with me this afternoon, my time, and morning, your time. It's been so fun to catch up with people and also meet new people and hear all of the cool things that are going on in your department. Um, Today, oh, sorry, was there a comment?
Not here.
Oh, okay. Sorry, thought I heard something. So today I wanted to sort of explore some previous and also some ongoing work on just one sort of sector of the lab, which is thinking about the evolution of reproductive isolation in the process of speciation. And so I certainly don't need to belabor the point that the world is incredibly speciose. So there are over 9 million eukaryotic species on the planet, and part of our job as EBers is to try and sort of describe this diversity, but also to understand why it evolves to begin with and what factors maintain diversity in nature. And in my lab, we're thinking of diversity largely at the level of species. Now, fundamental for our understanding of species level diversity are the formation of barriers to reproduction, which prevent two diverging lineages from collapsing back into one. These barriers have many different flavors. So, for example, they can manifest before a hybrid zygote is formed. We have a lot of really beautiful examples where differences in ecology prevent two lineages from really ever seeing each other in nature. And so they don't make hybrids. There are behavioral differences, which can prevent species if they do meet each other in nature. Perhaps they're not displaying the right courtship behaviors to solicit mates. And then, of course, there's a whole host of barriers that can manifest after mating has happened, but before fertilization. So, for example, incompatibilities between male gametes and female reproductive tracts. Our lab focuses a lot on barriers that happen after a hybrid zygote has been formed, in particular in these intrinsic postzygotic barriers. So problems of hybrid sterility or hybrid inviability. And I wanted to sort of pause here and note that I think the last maybe 2 decades or so of speciation research, sort of intrinsic postzygotic barriers have largely been, I don't want to say ignored, but maybe sort of downplayed in the literature because there was this conception that these barriers just take too long to evolve, and so they must not be important in the speciation process. But I want to sort of highlight that actually intrinsic postzygotic barriers, unlike ecological prezygotic barriers, these intrinsic postzygotic barriers, once they are complete, they're thought to be irreversible. And so they might play a particularly important role in sort of finalizing the speciation process or really creating sort of concrete measures of speciation. Also want to just sort of approach this question of do they take too long to evolve and say that actually it doesn't seem to be the case that they do take sort of too long to evolve to matter. So here what I'm showing you is the evolution of, in this case, it's hybrid sterility across genetic distance. This is data from helianthus or sunflowers from Greg Owens, originally from Greg Owens. And what we can see is that here, as genetic divergence accumulates, so each of these points is a pair of species, we can see there's a general increase in reproductive isolation. That's a classic finding, something we've known about for a really long time. But I want to point out that actually this relationship is incredibly messy. So there are species pairs that are, relatively speaking, quite divergent, that have very low levels of hybrid sterility. But on the flip side of this, there are also species pairs that are, relatively speaking, quite recently diverged and are either completely or nearly completely isolated through hybrid sterility. And this is true not just for sunflowers, but this is true actually for pretty much every group of organisms that we've looked at, with perhaps the exception of these fish, in which there's no sort of early diverging lineages that have strong reproductive isolation. And so I think when what's also sort of exciting about these data is that the messiness of them actually might be quite informative for looking at why these barriers evolved to begin with. And so that's a big sort of crux of the lab. We're really interested in trying to understand the different evolutionary forces that contribute to reproductive isolation. And also we're interested in trying to understand the genetic basis of reproductive isolation, how those alleles interact in hybrid backgrounds to make hybrids do so poorly. So although this is sort of broadly what we're interested in the lab, today I'm just going to focus specifically on one type of reproductive barrier, and that's the evolution of hybrid seed inviability. So you take two angiosperm species, you cross them, and the seeds don't germinate. The seeds are dead. And its potential evolutionary driver, which is this idea of parental conflict, which I'm going to sort of circle back to in just a moment. So like other intrinsic barriers, hybrid seed inviability is very common, and also it can evolve very rapidly. So here what I'm showing you is just the evolution of hybrid seed inviability on the right-hand side relative to another barrier, post-mating prezygotic. So that's that barrier that happens after mating, but before the hybrids are formed. And it's for three different groups of species. So Mimulus on the top, this is Holtomata in the middle, and then Streptanthus at the bottom. And what we can see for all three groups is that hybrid seed inviability seems to evolve more quickly than post-mating prezygotic barriers. And at least it achieves a higher rate of reproductive isolation more quickly. So this is, we think, a pretty important barrier in natural plant communities. And today I'm going to tell you about the role of parental conflict in its evolution. And so first I'm going to sort of make the bold claim that conflict is everywhere in nature. And actually it occurs at many different scales and in many different forms. So we can have conflict when the same individual has to house very different types of genomes, for example, cytonuclear conflict. We can have conflict when two different types of individuals have to share the majority of their genome. So for example, sexual conflict. And we can also see conflict manifest when selection acts differentially on a locus or a specific region of the genome relative to the genome as a whole, or individual level fitness. And that's been really nicely exemplified by, for example, these meiotic drivers. And it's been long hypothesized that these different forms of conflict can directly contribute to reproductive isolation and then to speciation, but we have few empirical tests, although they are sort of growing by the day. Okay, so the type of conflict that I'm going to focus on today is the conflict that arises between moms and developing offspring for how many resources to distribute to their kids. And so another sort of aspect of diversity on the planet is how many resources moms do give to their offspring. And that ranges everywhere from, for example, an elephant, which has like a 2-year gestation period, to, for example, maybe an externally fertilized fish in which maybe the eggs are sort of laid out in the reeds and sort of forgotten about. And specifically the conflict that I'm talking about today, I'm going to focus on a conflict that is mediated through some sort of intermediary tissue that facilitates nutrient exchange between moms and offspring. And so in angiosperms, that's the endosperm, and in eutherian animals, that's the placenta. And what's sort of special about these types of tissues is that they've actually evolved many times across the tree of life. So we often sort of associate placenta with placental mammals or eutherian mammals, but placentation type phenotypes have evolved, for example, in live-bearing fishes. And they also, there's some interesting examples of placenta or placenta-like features in skinks. In angiosperms, we have a really equivalent structure called the endosperm. And interestingly, there are some groups of angiosperms which have actually lost the endosperm. For example, orchids no longer have endosperm. And so when I'm talking about the conflict that takes place on these intermediary tissues, I'm going to refer to that as parental conflict. And because really, when I'm talking about conflict here, we're taking a gene's eye view, where we're thinking about the fitness of an allele when it's inherited through a maternal donor versus when it's inherited through a paternal donor. And so the conflict arises because maternity is guaranteed. So moms are equally related to all of the offspring that she produces. And so selection should favor sort of equivalent resource distribution among those offspring. But when there's any amount of multiple mating, dads are not equally related to all of the offspring that are produced within a litter or within a seed pod. And so you can get selection favoring the evolution of an allele that solicits more than its fair share of maternal resources. And so this tension between moms and dads can result in a coevolutionary arms race, where selection through dads is selecting for more and more resource soliciting alleles, and selection through moms has this sort of compensatory effect and selects for alleles that actually diminish resource or sort of resource repressive alleles. Okay, so how does this relate to speciation? Well, the idea is if you were to go out into any sort of given population, you might not find any evidence of this conflict. And that's because theory dictates that moms and dads should be pretty equally matched within a population. However, if you have different lineages that have experienced this arms race either at different rates or in different trajectories, then it's possible that maybe their hybrids will be a mismatch between this maternally repressive and paternally excessive set of alleles. Okay, so I just said that lineages might be experiencing this conflict at different rates or in different trajectories, and you might be wondering, well, what is driving that? And really the name of the game here is thinking about the variance in paternity within a fruit pod or within a litter or across years. And so typically sort of in the world of plants, we often have conceptualized this in the context of mating system, where we're contrasting, for example, lineages that are highly selfing. And so that individual acts as both the mom and the dad to all of the offspring produced, with a lineage that's highly outcrossing. And so when you contrast these two mating systems, you can imagine that outcrossers have increased variance in paternity, and so that favors stronger selection through parental conflict relative to a selfer, where those maternal and paternal interests are essentially aligned because it's the same genome, and all of the offspring are completely sort of identical clones of each other in a selfer. But there are other factors that might influence the variance in paternity that are perhaps a little less explored. So, for example, differences in demography, like a bottleneck, might reduce the effective number of fathers in a population. In plants, we might also think about life history in the context of clonality. So maybe a more clonal population might also have fewer effective fathers in the population. And we'll sort of circle back to this idea later in the talk. Okay, so one more thing you need to know before we really jump into the data, and that is that angiosperms are actually a fantastic system for studying parental conflict. And that's because of very specific way in which they're fertilized and which seeds develop. So when a plant seed is produced, what has to happen is pollen has to land on the stigma of the plant. It has to germinate all the way down that stigma, find the ovary, and then it partakes in two independent fertilization events. And so the first fertilization event pollinates or fertilizes this egg cell. That becomes the developing embryo and eventually the plant. But the second fertilization event has to fertilize this thing called the central cell, which is actually a diploid structure. So when it's fertilized, it becomes triploid with 2 parts mom to 1 part dad genomically. And it turns out that that 2-to-1 ratio is incredibly important for normal seed development because many genes which are expressed and essential for endosperm development are imprinted. By imprinted, I just mean that there is non-random expression of an allele, and that depends on whether it's maternally or paternally inherited. And it's the balance of these maternally expressed and paternally expressed alleles that allow seeds to develop normally. And we know that sort of imbalance of these megs and pegs can cause things to go awry because we have decades and decades of research in which people have twiddled with that 2-to-1 ratio by doing interploidy crosses. And overwhelmingly what we see is if you take, for example, a tetraploid and you use that as the maternal parent, so we have too much maternal expression, what we overwhelmingly see is that the seeds are much smaller on average, but also if we look at their development, we see that the endosperm has this sort of repressed or precocious phenotype where it doesn't get very big and it seems to get eaten up very quickly by the developing embryo. In contrast, if we now take our tetraploid and we use it as the paternal donor in this cross, we see that the seeds tend to be on average much larger. And the endosperm has this, what we call a sort of chaotic development. So it seems to get really big. There's some problems with cell division that happen there. And this direction of the cross is often, although not always, lethal. Okay, so you might be looking at these patterns and thinking like, wow, conflict is everywhere. These are perfectly aligned with parental conflict. Yep, you've just explained speciation in the first 10 minutes of this talk. And although I would like to take credit for that, that's probably not true. But what I'll just point out is that you can get these phenotypic effects with no sort of genotypic evolution. They can occur if you just take the same genotype and double its genome. You have these sort of imbalance of expression, and you'll end up with undergrowth and overgrowth phenotypes. But at the beginning of my talk, I told you that actually hybrid seed inviability is very common. And in fact, it's very common among diploids. So diploids that have accumulated these genetic differences and have not undergone a whole genome duplication event. And so this raises the question then, in these examples of hybrid seed inviability among diploid lineages, do we see that divergence at these normally imprinted genes are causing hybrid seed inviability? And are those alleles evolving through some process like parental conflict? And so these are the sort of questions that I'm going to try to tackle for you today. I'm going to first sort of outline the patterns of hybrid seed inviability that we see in this group. Then I'll spend quite a bit of time thinking about whether the patterns of hybrid seed inviability that we see conform to predictions under parental conflict. We'll briefly foray into thinking about the genetic basis of hybrid seed inviability, whether that aligns with a role of imprinted genes. And then lastly, in the last few minutes, I just want to sort of briefly touch on this idea that I've been brooding about and thinking about what are the consequences of hybridization in the face of really strong reproductive isolation. Okay. So this is the group that our lab works on. This is the genus Mimulus. It's a hyper diverse group of wildflowers. Of course, I don't need to belabor this to anyone in this audience, since you have so many great examples of Mimulus researchers in your own department. But it's very well known for these sort of beautiful wildflowers, really diverse in form and function and pollinator activity. But the group is also absolutely littered with intrinsic incompatibilities. And so here on the phylogeny, what I'm showing you is just the incidence of strong hybrid seed inviability. Those are shown in blue. And you can see that actually hybrid seed inviability is sort of sprinkled across this entire group, suggesting that it's actually evolved many, many times. If we zoom down here into the Guttatus species complex, this is the group that I work most on, what we see is that actually it's evolved many times within this group. Okay, so this is the Guttatus species complex. Again, it's a hyperdiverse group of very closely related species, probably sharing a common ancestor about 500,000 years ago or so. And again, it's well known for these really extreme examples of adaptation, either to, for example, toxic soils or high elevation, real diversity in mating systems, life history, all these types of things. Today, I'm just going to focus on sort of two of these species, something that I'm going to call Mimulus guttatus, which, as we'll learn, is a little bit of a catch-all for many different ecotypes, but I'm going to refer to it as one species, and I think you'll see why that is in just a moment. And I'm going to contrast Mimulus guttatus with this thing called Mimulus decorus, which at the beginning of my PhD, Mimulus decorus was just simply described as a morphological variant of Guttatus based on some kind of subtle stem trichome features. And we didn't really know much about it, including whether it even had any reproductive barriers. So one of the first things I did was I went out to the field and I collected several populations of Decorus from Oregon and Washington. We've now sort of expanded this sampling to cover most of its range throughout Oregon and Washington. Those are shown in gray. I've now also paired these whole genome sequences with a really rich data set that Alex Twyford and Janice Friedman created of Mimulus guttatus. Those are shown in the different shades of pink. And the different shades just denote these different life history strategies. So in tandem with the sequencing, we also completed a common garden experiment in which we grew about 50 populations of guttatus and Decorus in a common environment, and we looked to see whether there were any phenotypic differences between these named species. So what do we find? So here what I'm showing you is a PCA based on whole genome information. Again, these pink dots are guttatus, and the different shades of pink or red denote the life history. What we can see here is that actually, despite these, what we'll soon see as being very distinct phenotypes, genetically, they're quite homogeneous if you look across the whole genome. There are a few regions that are quite distinct between them, but on average, if you just sort of throw the whole genome into a PCA, they're quite homogeneous as a group. That's in contrast to Mimulus decorus, which I've now colored differently, and it's in this top right-hand corner. So you'll note that I've colored these points in blue, green, and yellow. And what this sort of shows is that in the process of this work, we discovered that decorus is actually a polyploid complex. So there are tetraploids, which are these green boxes, and there are these two distinct diploid lineages, one in yellow and one in blue. And we can see that Decorus on average is actually quite different from guttatus. We also did some back of the envelope coalescent estimates and estimate that guttatus and Decorus split about 230,000 years ago. So they're very recently diverged. Okay. So here's another PCA. This one is based on phenotypes instead of on genotypes. And really the main axis to pay attention to here is the axis of PC1 separating out these small and fast-growing annuals, that's shown here in the bottom right corner, from a large and slower growing perennial, which is shown up here. And what we can see is that although we get some nice separation out of annual guttatus, which is in this sort of bubblegum pink, all of the perennials are kind of all just jumbled right on top of each other. And in fact, our three different clades of Decorus, shown in blue, green, and yellow, are stacked all on top of each other and all on top of the most sort of perennial-looking perennial guttatus. And so what this suggests is that we sort of have a tale of two different types of species. We have Mimulus guttatus, which is phenotypically incredibly diverse, but these ecotypes don't necessarily have signatures of being particularly genetically different from each other across the whole genome. And then we have Mimulus decorus, which is phenotypically quite cryptic, but it is genetically distinct from guttatus. And we think that these two species separated about 230,000 years ago, so that's quite recent. So in order to quantify reproductive isolation, what I did was I teamed up with this fantastic undergrad at the time, Maya Wilson-Brown, and we crossed 19 populations of Decorus reciprocally to 4 populations of guttatus. And what we found, or maybe I'll just note that I'm only going to focus on the diploid crosses today. Again, patterns of hybrid seed and viability that we see in interploidy crosses can be attributed to that difference in dosage. And so we're going to just sort of ignore those for the moment. So if you take a diploid Decorus and you cross it to a diploid guttatus, what we find is that in one direction of the cross, the seeds are totally fine. They're nice and plump. These will germinate just fine. But the other direction of the cross is dead. So the seeds are flat. They look like they've been vacuum sealed shut, and they don't germinate. But what was very striking to us at the time is that the direction of the cross that yields a dead seed depends on what lineage of Decorus you've used in the cross. So when northern Decorus is the mom, or sorry, in crosses with northern Decorus, the seeds die when northern Decorus is mom. In crosses with southern Decorus, the seeds die when guttatus is the mom. Okay, and so we wanted to leverage this diversity of reproductive isolation to ask whether or not hybrid seed inviability was associated with parental conflict. And so in order to do this, we have to first make some predictions. So the first is that if parental conflict were driving the evolution of hybrid seed inviability, we should see signals of that maternal repression in one direction of the cross and paternal excess in the other direction of the cross. So our two reciprocal F1s should look different from each other. So we just simply measured seed size in all of our crosses, and this is what we find. We find that in one direction of the cross, the hybrid seeds are bigger than both parents, and in the other direction of the cross, the hybrid seeds are smaller than both parents. We'll also note that we also looked at endosperm development across seed development for all of our hybrids, and we see that pattern of precocious endosperm in the smaller seeds and sort of chaotic endosperm in the bigger seeds. And I won't show you those data right now, but I'm happy to chat about it if anyone wants to know. Okay, so next then, if these parent of origin effects on growth are actually causing seeds to die, then we should see that this discrepancy in F1 seed size is in some way correlated with the viability of the seed. And so here what I'm showing you is a relativized difference between our reciprocal F1s, where 0 means the F1s are the same. On the x-axis is the seed size, and on the y is their germination proclivity. And what we can see is that there's this strong relationship where if one F1 is bigger than the other one, then it's also much more likely that one of those seeds is going to die and not germinate. In this case, this pattern is driven by the fact that the seeds that are larger are the ones that are sort of flat and shriveled and don't germinate. Okay, so lastly, we would predict that if hybrid seed inviability was caused by parental conflict, that if we had some way of classifying the history of conflict that populations had experienced, we should be able to predict the outcome of crosses we hadn't done before. And so just to walk you through this sort of in a cartoon, if we think about conflict as a continuum, the further apart you are on this continuum, the more extreme those parent of origin effects on growth are going to be. And then the more likely it is that one or both directions of the cross will fail. And so in order to do this, what we did was we went back to our crossing data based on seed phenotypes, and we tried to make some predictions. So if we just look at our Northern Decorus first, what we see is that when Northern Decorus is the mom next to a Guttatus dad, we see that the seeds are overly large. And what this suggests is that those Northern Decorus maternal alleles aren't strong enough to deal with a Guttatus paternal allele. So those Guttatus paternal alleles are sort of winning out, and the seeds end up being overly large. And the flip of that is that when those Guttatus act as moms, they're also a really strong mom. So those Guttatus maternal alleles are too strong for a Northern Decorus paternal allele to sort of battle with, and the seeds end up being overly small. Now, if we contrast that with what we find with Southern Decorus, we actually see the opposite for Southern Decorus, where now the seeds are bigger when Southern Decorus is the dad, suggesting those Southern Decorus alleles are stronger than a Guttatus allele. And then sort of on the flip side of that, when Southern Decorus is the mom, the seeds are overly small, suggesting those Southern Decorus maternal alleles are also stronger than the Guttatus alleles. And so what this suggests overall is that Northern Decorus might have a weaker history of conflict than Guttatus, and Southern Decorus might have a stronger history of conflict than Guttatus. And so we think that these two lineages, even though they're sister to each other and they're genetically quite similar, we think they might have actually very different histories of conflict within their populations, with Guttatus being somewhere in between. And so to test this inference, we're going to do some new crosses. In fact, we're going to cross our two populations, our two lineages of Decorus, and we would predict under the framework of parental conflict that these two lineages should show the strongest inviability, stronger than either of them had with Guttatus. And so to test this, what I did was we performed another range-wide survey where we took a focal population from the north and a focal population from the south, and we just cross them to everybody. Again, I'm just going to focus on what we find within the diploids, but I'm happy to talk about what we see with the interploidy crosses as well. So if you take a population from the north and you cross it to all southerns, or you take a population from the south and you cross it to all of the northerns, what we see is that hybrid seed and viability is actually complete or nearly complete. So now it's pretty much no viable seeds are made in either direction of the cross. And in fact, if you measure seed sizes in all of these crosses, we again see these parent of origin effects on growth consistent with conflict. So the seeds are bigger when southern Decorus is the dad. Now you'll note that in this case, both F1s are now smaller than both parents. And part of this might just be because these seeds die really early on in development. So they might not sort of achieve their full size. Okay. So we see that we do have this sort of predictive power among diploids. But I want to go back to this continuum that I showed you just a moment ago. And I want to point out that actually this is quite odd. So here we have a situation in which we have two lineages. They're each other's closest relative. They're phenotypically almost indistinguishable. Their ranges abut in nature and are like 50 miles away from each other, Euclidean distance if you could fly through some mountains. And yet they seem to have these very different evolutionary histories. And so we wanted to sort of assess whether any of these different factors that could influence the variance of paternity were at play. What I'll just say is that both of these lineages, the southern and the northern Decorus, are both very highly outcrossing species, or at least they have the morphology of an outcrosser. They make big flowers. They have quite a distance between their stigma and anther, whereas in a sulfur, we'd expect those to be sort of sitting right on top of each other. And so we wanted to sort of assess whether there were these other factors, either demographic or life history differences, that could influence these histories of conflict. And so while we would love to do some sort of boots in the mud's ecology and look at demography in nature, we haven't been able to do that yet. And so instead, we're just going to rely on signatures across the genome. And so what I'm showing you here is pi at fourfold degenerate sites for all three of these lineages. And what we can see is that our weaker conflict species, the northern version of Mimulus decorus, does seem to have a much lower pi, suggesting that maybe it's experienced a bottleneck or is a slightly more clonal species. And so perhaps that's contributing to lower diversity, but also contributing to sort of a weakening of selection through parental conflict. On the other side of the equation for the southern Decorus, we see that it's roughly has the same amount of diversity as Guttatus. We're still trying to sort of suss out what might be going on in this species. It's a much more complicated story, it seems. But just to wrap up this bigger chunk, what I've shown you is that Decorus and Guttatus are pretty strongly reproductively isolated through hybrid seed and viability. We do see these patterns of parent of origin effects on growth, consistent with the role of parental conflict. We can infer a history of conflict and then predict the outcomes of subsequent crosses. And in this system, our inferences of conflict don't seem to be associated with a transition to obligate selfing, but might in other ways scale with diversity. And so just as sort of a quick plug, we've actually sort of continued on this path of thinking about the predictability of hybrid seed and viability. And so in my PhD, when I did these crosses originally, we picked these 4 populations of Guttatus, assuming that they represented all of the diversity that we could possibly imagine Guttatus holding in the context of hybrid seed and viability. And I will just tell you that I was wrong on that front. And so we've recently sort of re-upped the survey, and now we've done crosses involving 30 populations of Guttatus, crossing a full diallele cross. So all 30 populations cross to all other 30 populations in both directions of the cross. Just to summarize what we find from these crossing data, we see is that there's this population or series of populations that are in the Sierra foothills, shown here in this sort of deeper color of blue. And when you cross these Sierra lineages with either a coastal Guttatus, so as the name suggests, coastal Guttatus are growing on the coast, or a northern version of Guttatus up here in teal, what we see is that we see patterns of hybrid seed and viability, and the seeds are often, although not always dead, when either coastal Gutatus or that northern lineage of Gutatus acts as the mom, and those Sierra lineages act as the dad. Intriguingly, if you just sort of look at these seeds, we see these patterns of overgrowth and undergrowth as we saw before. So the overgrown seeds are the ones that are dead. And so actually just based on this sort of preliminary observation, what we might predict is that our Sierra lineage is also sort of a higher conflict lineage. And so we can apply our previous thinking and do some more crosses and see if we can't predict the outcome. So we crossed our Sierra lineage to both the northern version of Decorus and the southern version of Decorus, with the expectation that hybrid seed and viability should be very strong with the northern version of Decorus and much weaker with the southern version of Decorus. And so if you cross the Sierra lineage to the northern Decorus, what we see is that, again, hybrid seed and viability is complete in this direction of the cross. However, if we cross the Sierra lineage to our southern Decorus, what we see is that we can actually restore seed viability. So these seeds are perfectly happy and they germinate just fine. So we're excited to dig into this a little bit more, but that's all I have for you today in the context of these sort of patterns that we're seeing. So next, I wanted to sort of briefly touch on some of the work we've been doing trying to understand the genetic basis of hybrid seed and viability. And so typically when we think of the evolution of incompatibilities, we invoke the sort of Bizum-Dobzhansky-Mueller model in which derived alleles at two or more loci are fine in the backgrounds of which they've evolved, but there's something about their combination that makes things go awry in hybrids. We have a bit of a twist in hybrid seed and viability. These reciprocal F1s are genetically almost identical, and yet they're phenotypically very different. One is dead and one is alive. And so what I'll also just note here is that we have done a series of crosses to show that the patterns of hybrid seed and viability that we see cannot be explained by a cytonuclear incompatibility. And these plants don't have sex chromosomes. So those are usually sort of the typical go-to perpetrators of an asymmetric incompatibility. But there is a third model, and that's a model of nuclear alleles that have a parent of origin effect. So maybe it's the case that seeds are dead only when you inherit the big A allele from mom and the big B allele from dad. Any other combination is totally fine. And this is a model that's consistent with imprinting. So in order to sort of map these loci and look for nuclear loci with parent of origin effects, what's great about this group is that we can do these crosses sort of in all pairwise directions. Today, I'm just going to focus on what we have for hybrid seed and viability between Gutatus and the northern version of Decorus. So to do this, we created an F1 hybrid. We backcrossed it to both parents in both directions. And what's nice about this is this creates 4 different mapping populations. And because we've used the F1 as both the mom and the dad in these crosses, we can disentangle these parent of origin effects. And so what I'm showing you in the proportions below is the proportion of viable seeds. And you can already start to see that there are these parent of origin effects. So it matters who was mom and who was dad in this cross. And so we can actually map this, and we're going to use we're going to use segregation distortion mapping in order to achieve our goals. And to do this, what we do is we take all of the seeds that we have from these crosses and we just throw them on soil, and then we sequence who survived. And these survivors are a non-random sampling of the 2 parents that created them. Because by nature of the fact of being alive. So they will have a deficit of alleles that kill you. And so we can just march along the genome and we can look for regions of these segregation distortion or distortion away from Mendelian expectations. And specifically, we can compare our 2 directions of the cross to look for regions of the genome that when inherited from one parent have strong signals of segregation distortion, but have no such patterns when inherited from the other parent. So a parent of origin specific segregation distortion. And so just to show you what we found so far for our back crosses towards northern Decorus. So in this direction of the cross, we're looking for Gutatus paternal alleles that are associated with death. We find 5 such regions of the genome, just to show you what these actually look like. I'll zoom into chromosome 13. So this is the direction of the cross where we don't expect to see any segregation distortion, and indeed we don't really see anything going on there. However, if we look at the reciprocal direction, so now using the F1 as the dad, what we see is that the whole chromosome is distorted with this very big peak at the end. Intriguingly, under this peak is a gene called FIE, which is actually a master regulator of imprinting in angiosperms. Also just note that these QTL are very large. There's hundreds of genes under them. And so we have a lot of work ahead of us to try and narrow down these QTL interval, but we're excited about some of the candidate genes that we're finding. We can also take this analysis one step further, and we can ask, how bad is it to get a Getatus allele paternally versus inheriting the exact same allele maternally? So what is the parent of origin selection coefficient? And it's massive, as it turns out. So it's really bad to inherit a Getatus allele paternally, given that you have decorous maternal alleles at the rest of your genome. Okay, so that was the paternal side of the cross. We can also use our back cross towards Getatus to disentangle the maternal side of the cross. So what are the decorous maternal alleles associated with death? And when we do those analyses, we find 3 such loci. And again, they also have very strong selection coefficients. Okay. So as it turns out, I'm not the only person who's been working on hybrid seed and viability, and I'm not even the only person in Mimulus. And so really awesome work coming out of Andrea Sweigart's group at Georgia and Elin O'Neill, who's a researcher at Duke, have also been looking at hybrid seed and viability in other species in this complex. And I wanted to just compare the QTL that I've been finding with what they have found. So here what I'm showing you is a QTL map that Andrea published a number of years ago now. And I'm just doing the sort of qualitative assessment of looking at broad QTL regions that are shared either between what I've just shown you and what Andrea is showing, those are in blue, or between all 3 studies, and those are in red. And what I want you to take home here is that actually there are more QTL on this plot that are shared than there are that aren't. And every single QTL that I just showed you is on this map, except for one. And so I don't think that this is any sort of smoking gun evidence for the repeated evolution of hybrid seed and viability at the genetic level, but it's something that we're becoming really sort of increasingly interested in. And I think just sort of broadly as a field, we thought a lot about how repeatable evolution can be when it's caused by ecological selection. And I think we're sort of just beginning to scratch the surface at how repeatable evolution can be when it's caused by these more sort of conflictual processes. So just to wrap up this part, what I've shown you is that the genetic basis of hybrid seed and viability is complex, and it does involve these parent of origin effect alleles consistent with the role of imprinting. This is something we're really excited to dig into further and actually sort of measure imprinting in terms of expression, look at divergence in imprinting between our species, and then see whether these divergently imprinted loci are actually under our QTL peaks. What I've also shown you is that selection against these alleles is very strong and parent of origin specific. And then I sort of hinted at the potential for genetic parallelism. Okay, so just in our last 5 minutes or so, I want to briefly touch on some more recent work thinking about the consequences of secondary contact when 2 species with strong reproductive isolation meet each other again in nature. And so when we think about the diversity of life on Earth, of course, the evolution of reproductive isolation is really only half of the equation. Once a species forms, it also has to persist. And of course, there are lots of different factors that go into species persistence, but one that has become sort of increasingly more common to think about in speciation world is introgression, or the exchange of genetic material between these species. So intriguingly in this group, what we find is that decorous and Getatus actually co-occur quite frequently within their range. And so back before the pandemic in 2019 as a postdoc, I was able to go back out to the field with another awesome undergrad hope and we collected about 300 maternal families from 5 contact zones between Guttatus and Decorus. These are the southern version of Decorus. And what we did was I brought those seeds back to the lab, back to UNC where I was a postdoc. I wrangled some fly geneticists who were working with us to help me plate out subsets of seeds from every maternal family that we collected to estimate how many inviable seeds were being produced in nature, to try and estimate rates of hybridization. And then we grew a subset of the survivors from these plates in the UNC greenhouses. And although I won't talk about these today, we're actually like next Tuesday, just about to submit all of the DNAs from these plants to the Yale Sequencing Center to try and get an estimate of introgression in nature. Okay, so I don't have those data for you today, obviously, but I will just briefly touch on what we're finding in the context of how many inviable seeds are produced per mom. And so here I'm just showing you those data for one of these sites. This is the proportion of inviable seeds made, and each point is a mom, and the error bars are just the variability among replicate plates. And what you can see is that it's tremendously variable. So there are some moms that make perfectly fine seeds in nature, no bad ones. And there are some moms that are making entirely bad seeds in nature. And then there's a lot of moms that are sort of in between. And so I think there's a lot of different things going on in these data, and we're sort of excited to try and decompose what might be happening, including the fact that maybe some of these moms are actually hybrids themselves. But I think what we can say pretty confidently is that there is hybridization happening between these species in contact zones. We also paired this with some of our genomes to estimate introgression. And so here what I'm showing you are just D statistics. In the red box, I'm doing this for sympatric pairs. So with southern Decorus and Guttatus on the top, northern Decorus and Guttatus on the bottom. And then the right-hand side, I'm showing you the same comparisons, but instead I'm using 2 allopatric accessions of Guttatus now. And what we can see is that the D statistics are significant and also significantly larger when we look at our sympatric accessions, suggesting that there is introgression, ongoing introgression in these zones of sympatry. But we don't see, largely, we don't see significant D statistics in our allopatric accessions, suggesting that this isn't, our significant patterns aren't caused by, for example, more complicated demographic history or maybe older introgression that's sort of hard to parse out. So this observation that hybridization is happening, introgression is happening, and also that hybridization is probably not great in the system because you're making a bunch of bad seeds. Those 2 observations together really sort of sparked, are there other sort of effects, negative effects of hybridizing, not just the direct consequence of making bad seeds, but are there indirect effects as well? And this really sort of stems from thinking about parental conflict. And so thinking about this idea of these paternally derived sort of greedy alleles. And so we might suspect then if you hybridize with a species and they act as the paternal donor, and they are passing along a lot of really greedy alleles or alleles that solicit a lot of maternal resources. Because maternal resources are finite, that might actually detract away from, for example, intraspecific seeds that you're also making. So you might have these indirect effects on pure species seeds when in concert with hybrid siblings. So to test this, what I did was another series of crosses where we took a lineage from, or an accession from each of these lineages. We self-fertilized it. Those are going to stand for our intraspecific controls, where self seeds are just developing along other self seeds. We're going to pair that with a series of mixed pollination crosses, where I took an equal amount of pollen from 2 species, I mixed it together, and I reapplied it back to those same 2 species. And in this system, because the hybrids are very noticeable, either because they're dead or they're really, really small and they actually have a very dark coat color, we can actually remove the hybrids and we can just look at intraspecific seed size or self-seed size when those seeds develop alongside other selves and when they develop alongside hybrid siblings. So for the sake of time, I'm just going to focus on one of these trios of crosses, and that's the one where Guttatus is the mom. And so here we have 3 contrasts. We have Guttatus selves, We have guttatus selves that have developed alongside hybrid siblings from a relatively weaker dad. And so we might expect those selves to be a bit bigger. And in contrast, we also have selves that have developed alongside hybrid siblings from a relatively stronger dad. And so we might expect those selves to be just a little bit smaller. And when we look at seed size across these three crosses, that's exactly what we see. So the seeds are a bit bigger when they develop alongside weaker hybrid siblings, and they're a bit smaller when they develop alongside stronger hybrid siblings. And so what this suggests is that there are these, there's the potential for these indirect effects in secondary contact zones, whether that's for example, a cost, so making smaller seeds, or potentially a very, very, very small benefit, at least to the individual seed, depends on who you're hybridizing with. And so just to wrap up, what I've shown you is that heterospecific matings appear to be fairly common, and we do see signals of introgression. We also have some evidence that there might be these indirect effects of hybridization in nature. And so in total, what I've shown you today is that parental conflict might play this dual role in speciation, first by directly influencing the evolution of reproductive isolation through divergence in imprinted genes, and secondly, through these indirect effects by indirectly influencing conspecific seed development when those conspecific seeds develop alongside hybrid siblings. And so with that, I'll just say particularly thanks to all of the awesome undergrads who helped with this work, to my lab, and also to Aiden for the invite. Thank you all so much.
Okay, so we'll try this with mic, and if necessary, I can repeat questions down below, but any questions?
That was really nice. I have a number of questions, but so here are two questions. One is a small question. So can you rescue the hybrid seed inviolability by applying some kind of chemical compound that removes DNA methylation during development? Like, have you tried to rescue that phenotype?
I'm so sorry to announce that it's quite hard to hear you. Can you repeat the question? Oh, thank you.
Can you hear me, Jen?
I can, yes, thank you.
Yeah, so I think the question was, Andy was asking whether you can, I maybe missed part of the question, was whether you can modulate the system by adding some kind of chemical compounds like, things that mess around with DNA methylation, et cetera. I missed the other part. And rescue that phenotype that way.
Oh, yeah. Yeah, these are great questions. So the answer is yes, actually. So what people have shown is that essentially if, how does it go again? If you rinse the parental seeds in a chemical solution that essentially dissolves all methylation marks, and then you grow those plants up and then you do the crosses, people have shown in other systems that you can actually rescue hybrid seed inviolability. So there does seem to be this relationship between methylation, which is for people in the audience who don't think about imprinting that much, that is one mechanism by which the imprints are made, are differences in methylation. So people have shown that, at least in a small number of systems. The other thing that you can do in angiosperms, which you can't do in mammals, is we can do what's called an embryo rescue. And so you can actually dissect out that embryo and you can put it on fake endosperm, like coconut milk, and it will grow into a regular plant that's totally fine. And so what this suggests is that the incompatibility that we see that kills the seed is actually one that's born in the endosperm, not in the embryo. Yeah, and I guess the chemical analyses really sort of suggest that there's some sort of issue of imprinted loci that act in a dosage-specific manner.
Okay, great.
That's really cool. Thanks, Jen.
Thanks, Andy. Good to see you.
Other questions? I can probably just relay them from the front of the computer. Oh, in the back. Want to yell your question or we'll try?
Yeah, whatever you. Yeah. The other one was a bit more hearable. So I noticed that in the phylogeny, when you showed it there were like a lot of lineages with long branches, and at the very end there were like these pairs of sister species connected by. Oh, is that not?
I think it's the setup on there.
Can you hear me
Yeah.
from here? Yeah, maybe the mic's sorry.
Sorry.
No, I just saw a very curious pattern in the phylogeny where there were like all these pairs of sister species connected by very short branches, and then like each pair was like rooted to the rest of the phylogeny by really long branches. And I was wondering if this was like common for plants or like specific to Mimulus, or if that's anything to do with the parental dynamics.
Oh yeah, that's a great question. The real answer is I would not interpret the branch lengths on that phylogeny as being meaningful in any capacity. It was a figure that I made as a grad student, just as sort of like a cartoon depiction of the relationships. It's not, so the branch lengths are meaningless in that phylogeny. But it is true that for the group, sort of broadly speaking, we see very short branches, but we also see like very short internode branches. It's just a genus that seemingly has a sort of a history of pretty rapid diversification. Yeah, sorry for the confusion there.
Thanks.
All right. And it looks like there's a question in the chat. I'm curious about the nature of how conflict and hybrid species incompatibility evolves in the system in the first place. Is there an extrinsic benefit to conflict that makes it adaptive? Or is this evolutionary Murphy's law? If adaptive, what might drivers be? Does the speed of its evolution tell us anything?
Yeah, it's a great question. So I guess there's sort of two answers. The first is, can I definitively say that there's nothing ecologically going on that would influence the dynamics of conflict? No, I can't. So it's quite possible that there are differences in ecology sort of outside of this mating system demography explanation that I was talking about a little bit earlier in the talk that might sort of favor differences in something about how the seed is made, and then that sort of manifests through conflict. But I really want to stress the point that actually for conflict to evolve, you don't need to have any ecological differences. By nature of selfish evolution, you will have compensatory evolution in the absence of all ecology. So that is true for meiotic drivers, for TEs, for cytonuclear incompatibilities. You don't need to have ecology. Now, of course, that's not to say that ecology doesn't make things more interesting. Or, for example, in the case of parental conflict, differences in mating system are really strongly associated with differences in the strength of parental conflict, because it dictates that variance in paternity that a given, for example, fruit is experiencing. So that's sort of how they sort of directly link, but in general, you actually don't need this sort of ecological divergence or anything. All you need in this case is just a bit of allopatry to get things going, so that these two lineages are able to experience conflict differentially and not swap genes while it's sort of ramping up, and then you're sort of on the route of an arms race.
Okay. Emily? Maybe it's easier.
Right here?
Yeah, just
Okay.
it's the
Yeah.
computer mic.
So I was wondering about the tetraploids, and I was curious about whether the tetraploids formed through a whole genome duplication, or if you could tell if they were hybrids between the two different populations, and what effect, if there was any effect that you thought, if there has been any impact on the incompatibility that you've seen because these are tetraploids.
Yeah, that's a great question. The real answer is that we actually don't know a lot about these tetraploids, other than they've experienced a whole genome duplication. But whether that's from a single species or through an event of hybridization, there are some hints from, for example, this PCA that they're allotetraploids. And some other analysis we've done suggests that they might be allotetraploids or hybrid polyploids formed through hybridization. But whether they're between these two lineages of Decorus or between the northern Decorus and, for example, Guttatus is unanswered. We have some hints that maybe it's between these two lineages of Decorus, but we're not quite certain. One thing I can tell you about the tetraploids, though, in the context of their cross compatibility, is that they cross like a southern Decorus. So when crossed to Guttatus, the seeds die when Guttatus is mom. When crossed to northern Decorus, the seeds are entirely inviable in both directions. And these tetraploids are actually completely compatible with southern Decorus. So other than that, we don't really know what's going on in these tetraploids. It's a pretty open question.
Okay, Andy's got one more.
I can't help myself, Jen. I got one more question for you.
All right,
So,
I'm ready.
so I was really struck by the fact that your talk came full circle at the end. At the beginning of your talk, you were talking about how, look, there's been a lot of attention on post-mating prezygotic, but I'm going to focus instead on hybrid seed inviability. And you have these beautiful data that show that this is so important. And then at the end, you left us with this nugget, which basically showed us that hybridization that's at least potentially happening in the wild is setting up the conditions for reinforcement.
Yeah.
So what's up with that?
Yeah, I mean, I guess the answer is like any intrinsic barrier sets up the stage for reinforcement, given that you have secondary contact and hybridization going on. So maybe I'll make the claim that I'm not special in that arena. We don't see any evidence of reinforcement in this system. Of course, there's always a million traits you could look at, and maybe we're just not looking at the right ones, but we don't see any displacement in flowering time. The flowers are very largely similar between allopatric. I'm putting allopatric in quotes here because the ranges of Decorus are nested within Guttatus. So there's no population where we could say with confidence that there hasn't been some history of secondary contact. We only know that, at least currently, when we go in the field, we don't see Guttatus there, but we do see Decorus or vice versa. So we don't see any evidence of reinforcement. Interestingly, though, in these contact zones, we also don't see any evidence of the barriers going away. So another sort of predicted outcome of secondary contact is either you go to reinforcement and speciation becomes complete, or at least closer to it, or the barriers can go away if they're not complete. And even these intrinsic barriers can also sort of erase through time if reproductive isolation is incomplete. And so we don't see any evidence of that either. Hybrid seed inviolability is very strong in the contact zones, just as strong there as it is in sort of allopatric crosses. And so that's intriguing as well. So I guess there's two sort of interpretations there. Either it's just too young, and maybe the system needs a bit more time to play out, or perhaps something else is going on. So maybe these hybrid seed inviolability is sort of a stable state, and these plants just make so many seeds that it doesn't matter if one year they make a couple of fruits that are full of bad seeds. That's not enough selection to sort of really select for reinforcement. But yeah, we don't know. It's a good question.
All right. I think we've gone slightly over time, so we should probably call it there, but there's going to be a virtual Zoom reception. I think Jen will stay on for after this. So it's in the same Zoom room. It's on the schedule that's sent out. So if you have more questions, I encourage you to sign on there in a few minutes. Okay. All right. Let's thank Jen one more time.
Thank you all so much.